So hi, hello and welcome, Microbehunter here, and today I would like to do a special request video. I received an email from a very young biologist. Here is the email, and today in this video I'd like to explain to you what DNA is, and of course because this is a microscopy channel, I will be also putting a few cells under the microscope to show you where you can find DNA, and I'll explain also a little bit of some other background material. But let's first have a look at this email here. Hi there. I am 9 years old and I love biology. I always watch your videos, and I just wanted to ask you if you could do a video about DNA and what it is made of and what it does. I'm sending this message with the permission of my parents, and of course thank you very much for this email. It's always a very nice to hear that people are interested in biology, and of course also in microscopy as such. What I'm going to do now is I'm going to start off this video by asking you two questions. The first question is a rather simple one. It's ridiculously simple, and the second question is quite difficult, and these two questions will lead us directly into the topic of DNA. And then of course because after all this is a microscopy channel as well, I'll be also showing you how you can prepare some cells from the body so that you're able to see where you can find the DNA. Well, let's have a look here first. What I've got here is a few animals, and there are two. These are supposed to be sheep, a mother and father sheep, okay? And those two sheep, let's say they have babies, right? And I'm going to now start off with the easy question. I mean, look, I've got a butterfly here, over here, another sheep, a rabbit, over here, and this here, that is a so-called a moss piglet, or also known as a tardigrade or water bear. They have 8 legs and they're microscopic animals. And the first simple question is of those 4 animals here, water bear, rabbit, sheep, and butterfly, who is the child of mother and father sheep? Well, it's ridiculously simple. Of course, you're going to say, well, of course, it's the sheep. Sheep can only have sheep as a baby, obviously. But now here is the difficult question, the challenge. Why? Why is it that sheep can only have sheep as babies? Right? It's so obvious that essentially there cannot be any other animal, that we never really questioned this, right? But DNA is responsible for that, because it is actually DNA that is responsible for passing on the information of what it means to be a sheep from generation to the next generation. And in order to understand this, how this actually and where this information is this is something where we can then look under the microscope to actually have a look at the so-called the nucleus of a cell, because this is the place where the DNA can be found. But let's put all of those animals, including the butterfly, aside for once, because I've got something else that I want to show you here. Another little thing that you are already familiar with. Of course, they are watching what I'm doing here. This is of course a tomato, and Let's cut it. Let's cut it open. I'm not going to put it under the microscope, but I have something else to show you in here, because in here, of course, those little things here, these are the tomato seeds, right? And let's take out one of those here. Let me see if I'm able to manage that. Okay, here is a tomato seed, and I'm going to now use some tissue paper here. Okay, and let's remove the pulp of the tomato seed. Okay, so all you have to do is you have to kind of move it here and here. This is now the tomato seed, and it's difficult to catch. Here it is. Okay, right. And now if I put the tomato seed into the ground or on a wet tissue paper for a few days, a new plant is going to start to grow. And I'm going to ask you now the same question: which type of plant is going to grow out of this seed? Well, if you're going to say, well, of course, a tomato plant. Yeah. And I'm asking you, why a tomato plant? Why not a lemon? Okay. How does the seed actually know that it's supposed to make a tomato plant and not a lemon? Yeah, and here the answer is the same because the seed has DNA in the cells that tell it to form a tomato plant. Yeah. So again, here, just like with animals, the DNA is responsible for passing on the information of what it means to be a tomato plant from generation to generation. Yeah? So of course, I don't want to waste any tomatoes, so I'm going to eat it. Tastes good. I like it. I like it a lot. Okay. So, but I would like to do now the following. Let's have a look at a question here. Can you eat DNA? What a question. Can you eat DNA? As a matter of fact, what do you think I'm doing right now? I'm eating DNA right now, okay? Because the tomato cells, each tomato cell contains DNA. And when I eat a tomato, I'm eating tomato cells. And then, of course, I'm also eating the DNA. So can you eat the DNA? Of course, yes, that's what we're doing every day. So that's question number one. Question number two. Who discovered DNA? Well, the structure of DNA, how DNA looks like, and I'm going to show you the structure later as well. The structure of DNA was discovered in the 1950s. Over 70 years ago by 3 people. Okay. I've got, as a matter of fact, I brought some pictures here. So Francis Crick, he was one of the scientists who discovered DNA. We also have Rosalind Franklin and James Watson. So 3 people were responsible for discovering the structure of DNA. It was a lot of work, really. I like this picture a lot, of course, because there is a nice picture of an old microscope on here. Yeah, so Rosalind Franklin, Francis Crick, and James Watson are known for having discovered the structure of DNA. And Francis Crick and James Watson, they actually got also a prize, the so-called the Nobel Prize for having discovered it. But actually, Rosalind Franklin, she was also very important in discovering the structure here. So it's always important to remember the people who have spent so much time and so much effort in creating the knowledge that we now have. Okay, so this is basically question number 2. And the third thing that I would like to do now is I would like to now show you how you can find the cells in your body, and we're going to look into the cells to find the place where you can find the DNA. And in order to do that, what you need is you need a little Q-tip, as you call it, or cotton swab. It's actually for cleaning your ears, right? But they are very good also for collecting cells from inside of my mouth. So that's one thing that you need. Another thing that you need is you need of course, a microscope slide. The third thing that you need is you need some cover glasses. They're made of very thin glass. And it's always good to have some kind of a little container, because I'm going to be mixing some stain here, and this is where I'm going to start off here. Look, this is an ink cartridge. It's actually used for writing, so for fountain pens, and I'll be using this ink now to also stain the cells so that we're able to see the place better where we find the DNA. And look what I'm doing here. I have to squeeze this, and I'm going to really put a drop of the blue ink into this dish here, and it's too blue, it's too dark. So what I have to do is I have to add a little bit of water. So I've got a little water bottle here, okay, and I'll be adding a few drops of water as well, not too much, maybe about the same amount as I have ink, and then have to carefully mix it a little bit. So this is fine. This will do. And now it's time to collect some of my cells from inside my cheek, because we're going to now try to find the place of the DNA. So let's take the cotton swab and let's do the following. Look, I'm scratching the cells. Yeah, it's almost a little bit like brushing your teeth. And there are now on this cotton swab, there are now thousands of cells from my mouth. Okay, and what I'm doing right now is I'm just going to scratch it, not scratch it, but rub the cotton swab here on the microscope slide. Yeah, when you hold it a little bit up into the light, you're able to see that there is something on here. I don't know if you're able to see this. not so much, but actually it appears not to be so much, but actually there are many cells here. Okay, and I think there'll be enough. I'm going to throw this away, of course. Yeah, so there's a little trash can over there in the corner, and I'm now going to add a little bit of this ink water mixture. So what I'm doing is I'm taking a little bit of the ink here and I put it directly on, on my cells. Yeah. To put this away. Always be careful that you don't spill anything, because otherwise you have to clean up everything again. And I take one of those cover glasses, just make always sure that there's only one of them, and you put it on the ink. Ooh, and I already see that there's a problem. I've got too much ink, way too much ink. So what I have to do now is I have to take some tissue paper, and I'm going to remove some of this excess ink. You can see how the tissue paper soaks up some of this ink here. It's way too much. I do not want you don't want the cover glass to swim on the surface. So but I think that'll be fine. Okay, so I think this should be okay. And what I'm going to do now is I'm going to put it directly under the microscope, and I'm going to show you now some of my own cells. So I'm going to switch on the microscope view. So here, this is now okay. I have to switch on the light as well. And now let's have a look. I'm going to put it under the microscope now, and okay, what are we able to see here? It's a little bit blurry. Do you see all of those? Do you see all of those blue spots here? It's a little bit blurry, out of focus. Let me focus this here. Very nice, and I can already see 3 cells here. Let's find some more. All of those that you see here, these are cells. Look, I've got even an arrow over here. Okay, so that's a cell. It's all stained blue. That is a cell. That's a cell. That's a cell. But what I have to do now is I have to go up a little bit with the magnification, because what we want to do is we want to really look deeply into one of those cells here. So let's increase the magnification now. We always have to also go up with the light a little bit. Yep. So, and what we're able to see here is if you look very carefully, there in this cell here, there's this round structure. That is the nucleus, right? You see it's a little bit darker than the rest of the cell. And this nucleus, this is the place where the DNA can be found. Let's just to show you a little bit that pretty much every cell has those here as well. This cell is not so properly stained. There's one over here. Over here, we're not able to see it quite well. This round circle here appears to be an air bubble. Okay. Yeah. Here, you're also able to see it a little bit here. Yeah. That there is this oval structure inside the cell. Here you can see it as well. Okay. So let's find another place here. Here. Yep. Here is another cell. So you see that the cheek cells from the inside of my mouth, you also see are quite flat and thin, a little bit irregular in shape, but you can see again also here that this is this oval round structure. So this is the nucleus, and this is where you can find the DNA of the cell. Okay, and if you were now to analyze the DNA of my mouth cells, then it would be very clear and evident that this is of course human DNA. And not only that, if you have some kind of a comparison or reference, then you're even able to identify the person from who this is from. Here we've also got a cell, and you see that this one is not stained quite properly all the way, but you still see that there is a little round structure, the nucleus in here, right? And that is the place where the DNA can be found. In short, all living things have a DNA. Here as well, quite nice. The nucleus. All living things have DNA, because this DNA tells the cell basically what to do. It is the program of life, so to say. And to illustrate that also other living things have DNA, I want to just show you over here. These are so-called paramecia. These are single-celled microorganisms living in water. And you see that there are now 2 of them here. It used to be 1, but this one is dividing right now. So it's reproducing in that sense by cell division. And just in a minute or so, it's going to separate. As you see, it's almost separated here right now. And then they're going to completely separate, and then they're going to be 2 different individuals, right? And each one of those cells also has a nucleus, and it has DNA. So see, now they're really separating. Okay, now they're separated, right? So you started off with one cell, and then the DNA was copied, yeah. And here we're again at the beginning, and each one of those new cells also has, of course, the DNA of a paramecium. Yeah. So, and this is a very fundamental law of biology. Is that all living things have this DNA, and they use the DNA to pass on the information of life from one generation to the next. Yeah. So we just showed it to you first on the tiny little sheep, and then on the tomato, and now of course also on the paramecia here. And I also wanted to show you, of course, that also the cheek cells on the inside, they too have of course the DNA of a person. So I would like to move on now to the next question here. So let's have again the desk view here. What is DNA made of? Well, the answer is relatively simple. DNA is made of atoms, just like everything else in the universe. Every matter in the universe is made of atoms. Yeah. So it is a chemical substance. And this chemical substance is made of atoms, and yeah, just like we are made of atoms, of course, and DNA is, of course, also part of our own body. So, and we have another question over here, and this is the next question is now, how does the DNA look like? Well, that is what the three scientists have discovered. Remember, yeah, they discovered how the DNA looks like, and I've made a paper model, and so this basically means now we're going to really zoom in quite a bit. And look what I got here. Okay, this is a pretty long paper model, yeah, of DNA. So you can see it in large and in small. Yeah, here it looks kind of complicated, but if you look at it very carefully, you're going to see that yeah, it looks like a spiral, right? Maybe you're able to see it now much better here. Yeah, it's like a twisted ladder, and yeah, when you look into the DNA very carefully, then you're able to see that there are letters on the DNA: A, T, G, and C. So the DNA is made of 4 of these letters. Scientists call them bases. Yeah? So the 4 bases of the DNA have the letters A, T, G, and C. And these are the same 4 bases that appear in all living things. So all living things use the same 4 letters, but how are the living things now different from each other? Well, the living things are different because the sequence of the letters is different from living thing to living thing, from organism to organism. It's a little bit the same like in a book. The alphabet has 26 letters, but you can have many words and many more sentences and even many more books simply made out of the same 26 letters, simply by arranging the letters differently. So I just want to give you an example here. Here, for example, we have on one side TCT, right? TCT, and if this basically were the sequence in my DNA, then maybe the DNA of, let's say, a sheep is not TCT but TAT, for example. Yeah? So the sequence of those bases of those letters is different from living thing to living thing. And another interesting thing here in the DNA is that there are always 2 letters go together. So the A always goes together with a T, and the G always goes together with a C. Yeah. So if you know one side of the DNA, then you automatically also know the other side. So you can actually say that the information on the DNA is double, stored twice, yeah, on here. Now scientists call this type of structure here a double helix. A helix because it's twisted. It's like a ladder that is twisted. And the question that we now have is that's now the last question here is can you see the DNA under the microscope? And the answer is, well, actually almost not. I say almost not because the DNA actually is very, very thin. And we were only able to see the nucleus where the DNA can be found, but the actual double helix that we have here, strictly speaking, is extremely thin. And what you need is, if you have a very expensive and a very good electron microscope, then you are able to see the DNA as a very, very fine line. But generally, with light microscopes, this is not possible. Yeah. So the DNA, however, is quite long. The DNA is approximately 2 meters meters long in just one cell. So if you take one single cells that we've just looked at under the microscope, and if you take just the DNA of one of those cells here, of this nucleus, for example, and if you were to stretch it out, it's about 2 meters long. It's quite large, just of one cell. Okay, and it's able to fit in there because it's extremely thin. So it's very long and it's very thin. And in other words, this code that you have written, the ATGC code that you have over here, this basically tells the cell now what it's supposed to do and how it should work. Yeah? And it also tells the cell, if it's the cell of I don't know, of a tomato, of a lemon, of a sheep, for example, a butterfly, or of a human being. Yeah? So this is the program, essentially the DNA, that is essentially the program of life that can be found in all living things here, right? Yeah, and let's have a look again at one of those paramecia here. And one of the things that you might notice now is can you see those tiny little dots here in the background, right? Those tiny dots that you see swirling around here in the background, these are bacteria. And they are also made of cells, and they also have DNA. Yeah. Even if the tiny bacteria here in the background, those tiny little dots that you see swirling around, even they have DNA, because they too have to pass on the information from one generation to the next generation. Yeah. Now, if you want to see how I extracted the DNA, because it's possible also to extract and to purify the DNA, I did make another video. Well, I hope that you liked the video. Do please consider subscribing. Thank you again very much for your question. I think it was a very interesting and important question. But for today, I would like to say happy microbe hunting as always, and see you around next time. Bye bye.